Good morning. I would like to thank WAIMH for the great honor of being invited to speak here. And I'd also like to thank you for your graciousness in allowing me to present, being physically absent due to significant personal challenges. I deeply apologize for not being able to be there in person, and I'm grateful for your understanding. So the research in our laboratory asks an age-old question in psychology, which is how do we explain the links between early experiences that we have and our later emotional functioning? And for the purposes of this talk, I'm largely going to be focusing on emotional behaviors such as emotional reactivity, learning, and regulation. And as you can appreciate, especially in the case of the human, this link lasts a very long time. And therefore, there are many mediating pathways that we can investigate to understand this link. And the pathway that we choose to focus on in our laboratory is brain development. And so because I'm focusing on emotional behaviors like learning and regulation, I'm largely focusing on the connections between the amygdala and medial prefrontal cortex. Not because this is the only circuit that is involved in these behaviors, but they're certainly at the core of these functions. So the amygdala, of course, is this deep set of nuclei that are responsible for emotional attention and learning. And in maturity, we have these strong bidirectional structural and functional connections between the amygdala and the medial prefrontal cortex, which due to its cellular composition and its connectivity patterns is really well positioned for coordination and regulation of amygdala functioning. Now, when we think about the development of this neurobiology, particularly in the case of humans, we have to appreciate that it's not developing in isolation, but rather developing in a very special species expected context, which is the caregiver. And throughout this talk, anytime I refer to human caregivers, I'm referring to mothers, fathers, biological parents, adoptive parents, and so on, because the point that I'll be emphasizing throughout this talk is this relationship is a learned relationship, and I'm going to focus on the mechanisms underlying that learning. The other thing to point out is, particularly in the case of humans, we share some outlier status in the animal kingdom with regard to how long we spend in our caregiver's presence. So unlike most other species, we are spending time with our caregivers on the order of years or even decades, depending on the nature of your family. And this really begs the question of why Mother Nature would ever design a species like this, because it is metabolically very expensive to raise another human being for so long. And one of the answers that we find very compelling is that this long period of childhood affords us the luxury of extended brain plasticity. And during this time, the caregiver can provide very important scaffolding for our central nervous system development. So I want to spend a little bit of time talking about that exchange between brain plasticity and caregiving. So in many ways, the human brain is neotenous, meaning we retain these childlike characteristics for an extended period of time compared to other species. So if we take a species like a sea turtle, which does not receive caregiving, if this is developmental time on the x-axis, we see that in these animals, they take on adult-like characteristics, albeit smaller versions, but adult-like characteristics at a fairly early point in development. But as we move to more altricial species, we start to get this period of brain plasticity that exists prior to the onset of these adult characteristics. And that period of brain plasticity seems to be stretched out even further in the case of the human, where we have this tremendously long period of scaffolding by the caregiver, which might influence the developmental construction of brain regions like the amygdala and medial prefrontal cortex. Now, of course, when we talk about brain plasticity, it is a double-edged sword. On the one hand, brain plasticity, extended brain plasticity, might increase our risk for mental illness. So if this is age here on the x-axis, when we look at a number of mental health conditions associated with poor emotion regulation, they tend to have their clinical peak age of onset during this transition between childhood and adolescence, which is a stark reminder that these these mental health disorders are indeed developmental disorders. On the other hand, this extended period of plasticity is critical for our epistemic growth. So in other words, in order for us to go out and explore the world and gather information and become the incredibly sophisticated creatures that we are, we really need this incredible brain plasticity. Now, when we think about the drive to go out and gather information in the world, that drive exists in a very important and delicate balance with another drive. So during development, this drive for epistemic growth and exploration and the associated risk-taking underlying that exploration, that drive is kept in check by our great need for security and safety. So for example, this child here is able to explore his environment precisely by virtue of the fact that this species expected stimulus is present. So the parent enables efficacious exploration by being a source of security for the child. And so it's for this reason that Alan Strofe has written the caregiving relationship is an asset to the child in that it affords him or her the luxury of exploration and risk-taking, which is so critical for epistemic growth. And indeed, there's this seeming paradox that Ainsworth has written about, whereby our initial dependence on our caregivers actually gives rise to our ultimate psychological independence. And it's that balance which motivates a lot of our questions. Now, in maturity, our central nervous system plays a large role in ensuring our safe, independent exploration and necessary risk-taking. And today I'm going to be focusing on the amygdala and its connections with the medial prefrontal cortex and their central role in mature emotion regulation. And the premise of this talk is that the caregiving relationship plays a central role in establishing the neurobiology of emotion regulation. So what do we know about amygdala development? Starting with its volumetric development, we know that in humans at birth, the basic neuroanatomical architecture is already present. And there's this tremendous growth in volume in the first postnatal year of life. So the amygdala is anatomically well developed by the end of infancy, with some continued periods of moderate change throughout adolescence. But any of those changes are really dwarfed by what happens in this first year of life. And that's important to think about when we think about the experiences that are coincident during this first year of life with those big amygdala changes. Now, in our laboratory, we've been very interested in the functional changes that rest upon these early structural changes. And so we've relied largely on functional MRI. And what we and many others have found is, if this is age on the x-axis here, the amygdala seems to show some strong functional reactivity early in life. And that tends to attenuate with increasing age. And again, that strong functional reactivity seems really important to consider when we're thinking about early experiences coincident with this period of development. Now, when we look at connections between the amygdala and the medial prefrontal cortex, those connections seems to show a more protracted developmental time course, so that we may not see the emergence of adult-like functioning until adolescence, which has made this early period of development very interesting to us because the nature of the neurobiology is, or at least seems, so different than at points thereafter. And really begs the question, if connections between the amygdala and medial prefrontal cortex are so critical for mature emotion regulation, then what is helping provide that regulation early in life? And of course, one important part of the environment that's doing this is the caregiver. So we're interested in asking, what is it about this attachment relationship? It certainly promotes physical survival, but it also promotes some psychological benefits. What are the mechanisms of that bond, and what about timing? Why do primary attachment processes tend to happen early? So there's this quote from John Bowlby that I think is so important in some of his early work. He writes, in the countryside in springtime, there's no more familiar sight than mother animals with their young. So familiar are these sights, and so much do we take it for granted that lamb and ewe will remain together, that the questions are rarely asked. First, what causes these animals to remain in each other's company? And secondly, what function is fulfilled by their doing so? Now, this approach or affiliate behavior has often been thought of as being on an opposite end from avoidance-related behaviors. But indeed, it may be, as Bischoff has written, to understand the development of avoidance behaviors like fears, we must also understand the development of affiliative behaviors. Now, early work on the sensitive periods of emotional development have established that there's a very important sequence for the development of these two different learning systems. Namely, there's a maturational delay in altricial animals, that is, animals that require an attachment figure for survival. There's this maturational delay between affiliative learning systems, which come online first, and fear learning systems, which come on later. So in one example, here is a puppy. Puppies who are exposed to a human handler during a sensitive period for affiliation learning will exhibit an affiliation to that human handler, as shown here in this top image. However, for that same puppy, if we wait for first exposure to the human handler to occur at a slightly older age, that same animal will now exhibit fear or avoidance-related behaviors. And this ordering, this delay makes sense because fear learning interferes with affiliation learning. Affiliation learning is critical for attachment behaviors. And therefore, in a species who must attach in order to survive, we would want fear learning to hold off for a while so that affiliation can occur. Now, this ordering is so important that it will occur even in the presence of shock. Affiliation will occur even in the presence of shock, making this learning seem obligatory. And it's for this reason that Bowlby wrote, early affiliative learning seems to defy the rules of reinforcement learning, because affiliation will occur in early life, regardless of the valence of the stimulus. Now, more recently, Regina Sullivan's group has replicated this effect in rat pups using a fear conditioning paradigm. So young rat pups would be presented with a peppermint odor, which is initially a neutral stimulus and becomes the conditioned stimulus for these animals. This neutral peppermint odor is paired with a foot shock. And then that peppermint odor is placed on one arm of a Y maze and a control odor on the other. Very young rat pups will do what those puppies did with the human handler. They will approach that peppermint odor, even though it had been paired with an aversive foot shock. In slightly older animals, they will instead do what you and I would do. They will avoid that peppermint odor. They will exhibit that fear-related behavior. And it turns out that this same type of learning has been shown to underlie the learning of mother's cues and has been argued to be the basis of attachment-related learning, or that initial approach-related behaviors that's fundamental for attachment behaviors to emerge. And it turns out that the amygdala plays a large role in determining what type of learning is manifest. So I'm now going to show you those older animal data because at older ages, these are the data I just showed you, where those older animals will avoid that peppermint odor. At older ages, now having learned their attachment to their mothers, the mother's presence can now facilitate approach learning of new cues. So these cues become parent-affiliated cues. So what I mean is, if the animal is presented with a peppermint odor and a foot shock, when it's alone, it will do what you and I would do. It would avoid that peppermint odor. But now in this older animal, if its mother is reintroduced to the testing paradigm during acquisition, her presence will actually result in that older animal approaching that peppermint odor as it would have done at a younger age. So things associated with the parent become attractive. And that's what I'm showing you here, that older animal is approaching the peppermint odor when acquisition initially occurred in the presence of the mother. And the reason this is happening has to do with the amygdala. So here is rat age on the x-axis, and there's this period postnatally when the functioning of the amygdala is completely dependent on whether the mother is present in the nest or absent. So when the mother is in the nest, she keeps stress hormone cort low, which prevents the amygdala from engaging during fear learning. And so therefore, fear learning does not occur. Instead, the animal manifests approach-related behaviors. However, when the mother is out of the nest, those processes reverse, cort increases, amygdala becomes engaged during this learning, and therefore avoidance-related behaviors emerge. So when the animal is learning in its mother's presence, the amygdala is buffered, it's quieted, and approach-related behaviors ensue. When the mother is absent, this learning becomes amygdala and cort-mediated, and learning is manifests as avoidance behaviors. And these data demonstrate some important processes for attachment-related learning and give us some hypotheses to make about mechanisms underlying attachment-related learning in humans. These data are also an important reminder that the developmental state of the young altricial brain is not an immature brain, it's a brain that's actually perfectly designed to coordinate with parental input. So we've been interested to know whether similar types of learning occurs in the human. We would think that it does because humans also show attachment-related behaviors. So we collaborated with Sullivan's group and brought in young children. These were 3 to 5-year-olds. We would have gone younger if we could, but we have not done that yet. And children were presented with a conditioning paradigm. So the blue square was the condition, served as the conditioned stimulus, and that was paired with a aversive noise, so like loud nails down a chalkboard scraping. And the yellow triangle was never paired with anything. That was the CS minus. Children learn these associations either alone or in the presence of their parent. The parent was not doing anything, but the parent was just physically present during acquisition. Following acquisition, parents were removed and children were placed into a human Y maze, where the CS plus, that blue square, was placed on the door of one arm and the triangle placed on another arm, and children were asked to enter this maze and pick a door to retrieve a prize. And they were shown that it was the same bucket of prizes behind both doors, but we wanted to assess which door they were likely to choose. And what we observed was when children conditioned alone, they did more or less what you and I would do. They avoided that nasty blue square. However, when children conditioned initially in the presence of their parents, they did what those young rat pups did. Their learning switched to an attraction or an approach-related behavior, much like what was observed when rats conditioned in the presence of their parents. To ask what parents were actually doing during this learning, period to children's behavior, we modified a dot probe task. So here, children were presented with both cues, the square and the triangle. And then following presentation of those stimuli, there was a very brief slide of an asterisk, either appearing where the blue square was or where the triangle was. And the task was simply to press a button as quickly as one could to indicate the side of the screen where the asterisk appeared. And so we can use slower or faster reaction times to give us an index of where children's attention was on the previous slide. And we observed that when children conditioned alone, later on, they demonstrated an avoidance or a looking away to that CS plus. And this is consistent with what children do to fear-related stimuli in similar dot probe tasks. However, when children condition in the presence of their parent, instead, their attention was directed towards that CS plus. So there seems to be something changing at the level of attention that's influencing the way that children learn about these stimuli, even when the stimuli are associated with negatively valenced outcomes. To ask whether amygdala is similarly buffered by parental cues in humans, as was demonstrated in rodents, we used an fMRI paradigm where children were presented with blocks of pictures of their own parent and blocks of pictures of somebody else's parent, so a stranger. And we observed that during childhood, parental cues were effective in decreasing amygdala activity, which we interpret as parental buffering. At least at face value, it seems similar to what was observed in rat pups. But we did not see that same buffering during adolescence, which certainly doesn't mean that parents are no longer important in adolescence. They definitely are, but it may be that at the level of the amygdala, the parent has started to lose their efficacy in modulating brain activity. We also observed that connectivity between the amygdala and medial prefrontal cortex was strengthened phasically in childhood by presentation of the parental cues. So one idea that could come out of these data is that routine interactions with parents day in, day out during childhood may be part of what initially is phasic activity, but leads to strengthening of the neurobiology that will ultimately underlie emotion regulation. Now, the data that I've presented so far require that the parent themselves is calm and not distressed. But we know as effective as a calm parent is at buffering children's fear and stress physiology, a distressed or fearful parent is equally as good at potentiating children's fear and neurobiology. So to model this for better investigation in the laboratory, we examined parental transmission, not by fear conditioning children this time, but by fear conditioning the parent and then videotaping parents during fear conditioning. So here is an example. I'm the parent here, and I'm getting fear conditioned to this pink circle. Every time I see the pink circle, I flinch. I'm wearing headphones. I hear that awful noise. And we videotape that and show that video to my child, for example. And she will also observe somebody else's parent get fear conditioned to a different stimulus, a green diamond. And what we observe is that after acquisition, my daughter's amygdala, for example, would show greater reactivity to the stimuli that I fear conditioned to relative to the stimulus that somebody else's parent fear conditioned to. And not only would her amygdala be more sensitive to that cue, but she would learn to fear that cue better. So this is study phase along the x-axis, and higher scores means better disliking of a cue, or in other words, better learning. And while children can learn to dislike both cues, they learn to dislike the cue that their parent conditioned to better. And interestingly, there were some hints of individual differences here, such that this learning effect was potentiated by parental anxiety, not the child's anxiety, but the more anxious that parents were, the better children were at learning these associations. So we had the opportunity to not only scan children while they were undergoing observational learning, but we also scanned parents while they were undergoing firsthand conditioning. And we observed this intergenerational neurobiological correlation between the amygdala and prefrontal cortex of each dyad. So parents who exhibited greater amygdala reactivity while they were conditioning had children whose prefrontal cortex exhibited lower activity while observing, and vice versa. So these data tentatively paint a picture whereby we have an intergenerational association such that parents who strongly recruit prefrontal regulatory systems during affective learning may be indirectly able to buffer the amygdala activity of their children during observational learning. So taken together, these data are creating a framework for us where this is age on the x-axis. There may be this period early in life where parents are very able to increase and decrease fear-related neurobiology and learning, perhaps being having some privileged access to these deep emotional learning systems in the brain. And we have some ideas about the neurohormonal cascades that can ensue as a function of parent availability, or in terms of as a function of parent distress or regulation. And this idea here is what happens during this putative sensitive period may have longer consequences for the way that individual differences in emotion regulation emerges at later ages. And if so, this begins to give us some new questions to ask, or new ways to ask questions about development of emotion regulation systems following caregiving related early adversities, which I will abbreviate as CREAS throughout this talk. And when we talk about CREAS, we're referring to experiences that largely happen in infancy and can include abandonment or separation from parents, abuse, neglect, multiple caregiving switches, and or institutional care. So this is a heterogeneous set of experiences. And what they all have in common is the disruption of the parent-child relationship that we believe is ultimately critical in helping to establish the neurobiology of emotion regulation. So I'll now switch to talking about some of our recent data on development following CREAS. Now, we know from a large animal literature, largely from adults, already developed animals using a very different type of stressor, that stress can lead to changes in amygdala and medial prefrontal cortex. So in the amygdala, chronic stress, in this case, this was chronic immobilization stress, but chronic stress led to cellular hypertrophy and greater reactivity of amygdala cells. Whereas the opposite was likely to happen in regions of the brain like the medial prefrontal cortex. And I present these two in contrast to highlight that stress doesn't seem to have a uniform effect on the brain, but rather leads to adaptation in the brain, which presumably are designed to help the individual meet future stressors. Now, with these types of data, even though we're talking about a different species, different stressor, different age group, they generate some hypotheses for us about human brain development. Now, if we start off looking at broad brushstrokes behavior, we see that in terms of internalizing and externalizing problems, a history of CREs is associated with greater risk for both of these types of problems. And that's replicating many studies. What I'm highlighting here are the individual differences that we observe in these outcomes. And so we're very interested in interrogating what's contributing to these individual differences, despite the fact that CREs and similar behaviors are the leading environmental risk factor for mental health problems. So when we investigate what's happening at the level of amygdala and medial prefrontal cortex, we've observed evidence of amygdala hyperactivity, as well as atypical connectivity between the amygdala and medial prefrontal cortex. And when we further interrogate what the nature of this atypicality is, we were initially surprised by the findings. So as I mentioned at the beginning of this talk, children without a history of CREs, who I'll be referring to as the comparison group, these are just children who have always lived with their biologically related parents and have no reported evidence of any of these early adversities. In childhood, these are aged 4 to 9 year olds. The connectivity patterns between the amygdala and medial prefrontal cortex look quite different than they do at older ages, where we get what's a more regulatory pattern consistent with emotion regulation behaviors. However, following CREs, we see that in 4 to 9 year olds, the average response actually looks much more similar to older adolescents and adults. So these types of findings have motivated us to think about stress, early life stress, in this case, CREs, as potentially accelerating the development of this neurobiology. And although we were initially surprised by these results, because much of the framework in the brain development literature had suggested that early life stress might lead to delays in development, there's actually a large and growing list of findings from animal studies showing indeed that there are accelerations within this neurobiology. And I list just some of the findings here. Now, this notion that early life stress like CREs might accelerate development is consistent with some models posited by Janet Worker and Takao Hench that sensitive periods themselves may be plastic. That is, the timing of these sensitive periods may change as a function of experiences, and caregiving adversity may be one experience that may lead to precocious onset or precocious development of this neurobiology. And so the idea is that early amygdala hyperactivity, that's the result of these early stressors, may actually lead to the earlier instantiation of these adult-like connections with medial prefrontal cortex, which on the one hand could lead to some adaptive outcomes, and we have some findings suggesting that there might be short-term benefits for these putative accelerations. But at the same time, if true, this is also leading to a reduction in that developmental plasticity. And so we hypothesized that accelerated development following CREs may place limits on parental buffering of the amygdala. So we repeated that parent-stranger fMRI paradigm, where children saw pictures of their parents or of a stranger. And these are the data I showed you earlier, where children with a typical caregiving history showed what we interpreted as amygdala buffering. Following CREs, we did not see that evidence of that same buffering. And this is at the average level, of course, but what you may be paying attention to are the very large error bars here, which suggests that for some children following CREs, they're not showing this buffering. But for others, there may be evidence of buffering. And therefore, we interrogated those individual differences over longitudinal time to see if those differences were predictive of mental health behaviors. In this case, we're looking at anxiety. So if you pay attention to the right side of the screen, these are children with a history of CREs, and we split participants into those who showed buffering of the amygdala to their parent at time 1 and those who didn't. And at time 1, there were no group differences in anxiety. But by the time we saw them again 2 years later, group differences had emerged, such that those children who showed buffering at time 1 had significant decreases in anxiety, such that they were no longer statistically different from the children who were in the comparison group. Whereas those children with a history of CREs who did not show buffering at time 1 were those children who tended to maintain higher levels of anxiety 2 years later. Now, what was the difference between these two groups in terms of experiences? We observed that children who showed buffering of the amygdala at time 1 were also the group that were likely to report higher feelings of felt security or felt attachment security to their current caregiver. Note that these children had, all of these children had since experienced adoption and were living with different caregivers. And what these data are suggesting, at least, is the power of later caregiving relationships. These data may suggest that although caregiving experiences are important for everybody, they may be particularly important following CRE-like experiences. And those findings reflect other similar results in our laboratory, again, which are correlational in nature. If you pay attention to the right side of the screen here, these are children with a history of CREs. The higher the felt security children report feeling, the lower they're internalizing problems. Now, of course, these are correlational data, so we can't really say much about directionality, but I would quickly refer you to the work of my collaborator, Mary Dozier, who has used RCT designs for years, demonstrating that caregiving intervention actually moves these processes in a way that allows us to talk about causality. So in my remaining time, I want to present to you some of our newest work, where this research is going, because what's struck me for a long time is that this amygdala medial prefrontal circuitry actually emerges across a number of different behavioral domains. So on the one hand, this neurobiology is central to emotion regulation studies that have been conducted in adults. We and others have also shown that this neurobiology is highly modulated by parental cues early in development. This neurobiology, as I just discussed, is also shown to be altered or atypical following early caregiving adversity. And this begs the question, what is the common denominator here? And Roy and colleagues presented a conceptual synthesis suggesting that this circuitry may actually serve a broader function, which is to represent semantic level affective knowledge. And so we've been very interested in following this idea that this neurobiology may be doing more than just regulating emotions, which is sort of a process level function, that this neurobiology may actually support content level knowledge, in this case, affective content. And so I would like to demonstrate how that might work and some of the studies that we've been conducting to test this idea. So how might this content knowledge get built? I'm going to first use an example from cognition before I talk about affect, because the cognitive literature has been investigating this model for quite some time. So according to cognitive theory, first we have memories for discrete episodes. So for example, the first time that I met Dr. X, the first time I met Dr. X, I encode episodically that Dr. X had good insights in that conversation and was very kind to that student. Now, those episodic details are hippocampally encoded. But with time and repetition of meeting Dr. X and having similar episodes, these episodic memories become decontextualized, while the essential features of who Dr. X is, the essential content knowledge, is abstracted from and moved, transitioned from the hippocampus to these more midline cortical regions, including the medial prefrontal cortex, so that over time, I abstract the semantic meaning of who Dr. X is. So, for example, over time, I abstract the semantic knowledge that Dr. X is brilliant and compassionate. Now, more recently, we've been wondering if some similar type of process can occur at the affect, in the domain of the affect. So we've been wondering if, for example, the amygdala, which also encodes episodic memories, albeit affective and non-declarative in nature, may similarly be part of the process that underlies the development of this affective knowledge or affective schemas, such that what's initially learned at the level of the amygdala may over time become abstracted or semanticized to these cortical regions. So the hypothesis that we're testing is the development of amygdala cortical function actually reflects these non-declarative affective semantic memory structures. And might this framework get us closer to thinking about a neurobiological operationalization of internal working models? So in our affective example, an infant may have episodic encoding of I have a need and my need was met. Now that pairing may over repetition, time, and development become abstracted into the knowledge, the non-declarative knowledge that when I have needs, they are met. So might this neurobiology actually be representing that affective knowledge? So these early learning patterns help us make meaning out of new experiences or future encounters, make predictions about what's likely to happen at an affective level. So this theoretical lens may provide deeper insights into how attachment relationships help build the brain. So now we're asking, with this framework, what is actually that affective content? What's the content of these affective semantic memory structures following CREs? And in this case, the CREs that we'll be examining include abandonment or separation, institutional care, multiple caregivers, abuse, and or neglect. And to interrogate the affective content of these schemas, we collaborate with Simona Getty, who's an expert on memory development, and this is work led by my graduate student, Anna Vanucci. And we created this attachment false memory task, which I'll describe shortly. This was a pre-registered report with 160 school-aged children with and without a history of CREs. And children saw a narrative with built by a number of scenes. So in this example, a mother and child are playing together. They continue playing together. At some point, the child makes a bid to her mother. She's crying. And then there's this ambiguous ending. We don't know what happened. So children see many of these types of narratives built by scenes. There were actually 24 of these narratives. And in all of them, there's this missing scene. There's missing information followed by an ambiguous ending. After children watch all 24 of these narratives, we give them a surprise memory test afterwards. And we ask them whether they saw any of these scenes. Importantly, most of the scenes that we show them are false. They did not ever see these scenes. The two that we're interested in are these two. So one of the false scenes is a narrative that includes that's consistent with a secure internal content narrative. So the mother picks up the child who is making a bid to her, whereas the other scene is consistent with an insecure narrative. The mother is scolding the child for crying. And so what we're curious about is there an association between children's early caregiving experiences, whether they experienced CREs or not, and their likelihood of falsely inserting either one of these scenes? Now, the means on the right are going to represent children incorrectly inserting a secure scene into the narrative, while the bars on the left are going to represent children incorrectly inserting an insecure scene. So first, as you can see, there was no difference in falsely inserting a secure scene between children with and without a history of CREs. But there was an increased tendency of falsely inserting insecure scenes for children with a history of CREs, indicating to us that children with a history of CREs may have content knowledge in these affective schemas that is more consistent with insecure information. Now, to ask whether these schematized memories or this affective knowledge is represented in emotion regulation related neurobiology, we performed an fMRI experiment while participants viewed movies with attachment narratives. And the two movies we used were either The Little Princess or Homeward Bound. Now, these two movies differ in many, many ways, but what they both have in common is this narrative of the two central figures being together, followed by a significant separation, followed by a reunion. And this is work in collaboration with Chris Baldessano and led by Carolyn Lee and Sam Cohen. So we want to understand how the amygdala is functionally connected to other brain regions, for example, the medial prefrontal cortex, during this movie watching, which is a movie associated with attachment narrative. The typical approach is to measure functional connectivity using a simple correlation with the time courses of two regions within a single child's brain. So, for example, activity of the amygdala would be, or the correlation between the amygdala would be assessed with prefrontal cortex in an intra-subject manner. This is the typical means of assessing functional connectivity. Now, this kind of connectivity can be difficult to interpret because it can be driven by three different types of interactions. So there's intrinsic neural fluctuations that are unrelated to the stimulus and would be present even at rest, interactions related to the specific content of the movie stimulus, and also artifacts due to shared sources of noise present in both time series, for example, motion, when really all we're interested in is the stimulus-driven effects. So to better isolate these stimulus-driven effects, in this case, during the attachment movie, we can measure intersubject functional connectivity. Here, we take the time course of amygdala response in one child, and we correlate it with responses in other brain regions in all other children. This across-brain correlation zeroes out participant-specific noise. That is, the only way that these two time courses will be similar to each other is if they're time-locked, to information present within the stimulus. That is, this approach is analogous to the within-subject functional connectivity analysis, but it isolates the shared stimulus-induced signal and is therefore robust to idiosyncratic noise. And so we use this analysis while children are watching this narrative of being together, separate, and then reunited, and ask which regions share stimulus-related information with the amygdala during the attachment-related movie clip. So we first asked whether there were differences in connectivity during the attachment movie watching as a function of early caregiving-related early adversities. Now, this measure showed that the amygdala exhibited stimulus-related responses similar to those in early visual perceptual regions. That is, the amygdala is communicating more with low-level sensory information during this movie watching for those individuals with a history of adversity relative to those without. So in other words, caregiving-related early adversity was not showing us group differences between the amygdala and medial prefrontal cortex while watching these attachment-related movies. Instead, when we examined children's current feelings of caregiving security, we found that for all children, regardless of their early caregiving adversity, the amygdala in children with high attachment to their caregivers shared greater amounts of stimulus information with these regions in red, including medial prefrontal cortex. In other words, there was greater amygdala connectivity with medial prefrontal cortex in children with greater scores, higher scores on that felt attachment measure. And instead, children with lower scores on that attachment measure were more likely to show amygdala connectivity with perceptual regions like the amygdala, like the visual cortex. So these results suggest that the amygdala is driven more by low-level sensory information in children with low attachment security, and only the high attachment group used higher level knowledge about socio-emotional schemas when constructing their emotional response to the narrative. Now, this finding is very exciting to us because it suggests that the neurobiology that's been studied as underlying emotion regulation in maturity seems to overlap with the neurobiology that might represent attachment-related schemas. So perhaps this framework provides deeper insight into the mechanisms that link early caregiving experiences and individual differences in emotion regulation. So in conclusion, what we've been seeing is that the processes that are involved in attachment involve learning, and this learning may involve potentially unique neurofunction that's present early in life, involving the amygdala and medial prefrontal cortex. And the consequences of these relationships involve developing amygdala medial PFC circuitry that's part of the neural basis of our attachment-related schemas. So perhaps this is getting us closer to thinking about the neurobiology underlying internal working models. Caregiving adversity may alter affective content knowledge represented within this neurobiology of amygdala and medial PFC. And while strong parent-child relationships are always important, they may have especially powerful effects in the wake of adversity. And so our working model is that the neurobiology underlying emotion regulation is constructed by caregiving experiences, so that when we activate emotion regulation neurobiology in adulthood, we're actually reinstantiating the neural representations that were established by early caregiving interactions. So with that, I would like to thank the funding sources, lab members, important collaborators, families and individuals who participated in this work, and thank you for your attention.